Welcome back, and let's continue with understanding of biotechnology. We had an introduction already. These slides which you are seeing are something which I have made long time back, and initial certain slides here will give you an understanding of certain initial discoveries, all these introductory things which we are talking about, right? And I will share these slides with you. You can go through it, or you can go through anything else, any other reference book you go through. You will get the introductory understanding. Even Google and all will tell you what is biotechnology, genetic engineering, etc. Right? Just be careful about one thing. Like I told you, since biotechnology is a huge field, so there will be lots and lots and lots of information. Okay, so when it comes to examinations, whether your board or medical entrance examinations, you need to understand that the specific processes, etc., will be based upon what questions on the specific processes will be based upon what you have in the textbook, right? So like when we talk about pioneers in biotechnology, you can see we start right away from Anthony von Leeuwenhoek, who discovered bacteria using microscope. He's also known for discovery, or someone who has first observed live cells, because Robert Hooke observed dead cells. Then we talk about Mendel, who has first given some understanding on principles of inheritance. Louis Pasteur, who is well known for giving us lots and lots of understanding in terms of microorganisms. Okay, he is also often known as father of microbiology also. Then Robert Hooke, we already know. Watson and Crick, you already know, who gave us an understanding of DNA molecule. Bohlberg, from here we start talking about people who worked on technology associated with the DNA, right? So recombinant DNA technology or genetic engineering is the first time he has started certain methods by which genetic material can be inserted into one organism, right? Foreign genetic material, how to insert into an organism. So there are a lot of historical developments which you will be learning here by looking at these slides. You can go through it. 1983, for the first time, US Supreme Court has given approval for patenting of genetically altered organisms. So from then on, the society started accepting the organisms which are genetically altered, changed. Genetically, we call it today as GMOs, genetically modified organisms. And then there are a lot of GMOs which came into picture after that. Dolly, famous first cloned organism, okay, is also a advancement of biotechnology, right? Now, this entire chapter, as I told you, is divided into two parts. First is, what are the different tools that can be used for recombinant DNA technology or genetic engineering, right? Various tools, like how will you cut the DNA, how will you paste the DNA, how will you transfer the DNA, etc., etc. So those things, those tools we are going to discuss first. When we are talking about the tools, you may somewhere find it little difficult to understand that why these are required, right? So that is why let me give you a small brief of what exactly do we do in biotechnology. That is the second part of the chapter, actually known as the process. Okay, so that process I will first show you in brief. The various steps which are carried out. I am not going into details of this, but this is a basic understanding of what steps are carried out in biotechnology, in genetic engineering, or RDT, we often call it as. RDT stands for recombinant DNA technology. Okay, where recombinant DNA is also written as rDNA. So here you see, you can do one thing. In your mind, you keep some examples. Okay, when we try to understand with the help of an example, then it becomes easier. So I will just give you a brief history of how insulin was developed. Because today what you get in the market, insulin is one of the most common uh, biotechnology product that we have in the market. Okay, many of us are using insulin without knowing that we are actually using a biotechnology product. Okay, so it is one of the most common biotechnology product, most widely used maybe. So what happened is, when there was first understanding that this particular disease or this particular condition, which is hyperglycemia condition, is found in an individual because of deficiency of insulin. There are two reasons for hyperglycemia. One is insulin dependent, one is insulin independent. So insulin dependent is one where insulin deficiency is the main cause. So then scientists understood that to solve this issue, the only way out is to get insulin. So they have to introduce or inject insulin into the individual who has the deficiency. But then insulin is a hormone, and the hormones are not produced in large quantity in the body, right? Hormones are produced in very less quantity. So although there were technology available to obtain certain proteins from human blood, right? Like albumin is one protein. We know that albumin is that protein in human plasma which is present in very high or highest concentration. So from the voluntary blood donors, we collect the blood and we extract albumin from it. Okay, it's a fractionation kind of process which we have studied in chemistry, by which we can obtain the albumin fraction out of it. But insulin you cannot obtain like that, because it is hormone, and such hormones are produced in very small quantity, few nanograms maybe. So first approach was to collect your insulin from some animals. Okay, like bovine insulin was collected. Okay, and those insulin were very similar to human insulin. Those were given to human beings. It did work to some extent, but still, since it was a non-human protein, so it did produce certain immunogenic reactions. Okay, means the body did not accept it fully. So the next solution was that you have to get human protein only, human insulin only. There is no other way out. But you cannot get human insulin from human beings because it is produced in very less quantity. So what is the way out here? Then by that time, biotechnology was developed. So in very simple terms, if I say, what human beings did is, from a human cell, try to understand, from a human cell, they took out insulin DNA, okay, or insulin gene, right? So they took out the whole DNA, and from the DNA they cut out the insulin gene. Then this insulin gene was inserted where? Into a bacteria. Inserted into bacteria. Then what happened? Bacteria, it is very easy to grow in the lab. Okay, easily you can grow them. You can grow them in large quantities, very large quantities you can grow them. So from bacteria, what happened is bacteria started producing insulin. Okay, now bacteria is producing insulin, which is a human protein, but it is produced in bacteria. Now what you do? From these bacteria, you obtain insulin. Okay, in very brief I told you. From these bacteria, you obtain insulin now. Okay. Similar such processes are also carried out in other ways. There is one protein called as alpha-1 antitrypsin. Okay, and another one called as alpha-lactalbumin. Where these are produced, these are human protein which are introduced into cow. These are human milk protein. Okay, so when you were being breastfed by your mothers, at that time you got this particular protein from your mother's breast milk. But today, if you want to have that protein, you cannot have it. Why? Because your mother is no more producing breast milk. So where will you get this particular protein? Some of these proteins are having some other functions also. They are used for some medical purposes also. So where will you get this? You cannot ask a lactating mother to donate her milk, right? At the same time, lactation period is not unlimited. It's for a limited period of time that the mother will produce a milk. So what was done is this particular gene was introduced into cows. And now cow's milk started producing human protein. Okay. So in these all different examples, if we have these different examples in our mind, then it will become easier for us to understand the process. And once you understand the process, it becomes easier for you to understand that what tools are required. For example, here you say, we said that from human cell, you get the insulin DNA. Try to go into much deeper as possible. You try to go deep inside. Okay. Every single step, try to think on every single step how it is possible. In fact, that is how the research is carried out. So I simply said from human cell, you obtain insulin DNA. So can anyone tell me what will be the first step here to be carried out to get the insulin gene from human cells? Anyone can tell me what will be the first thing that you have to carry out? Come on, just think.
Separate the DNA from the rest of the cell.
Separate DNA from
The rest of the cell.
Even before that, you need to do something.
Separate the nucleus from the cell.
Before that, first thing,
Get one single cell.
is it possible to get one single cell? Do you think it is possible to get one cell? That is impossible. That is impossible. How will you get one cell? How will you work with one cell? You have some idea about the size of DNA and all. How is it possible to get one DNA? It's impossible, right? So your first step will be to obtain the tissue. Now the question is, from human being, you need the tissue. Which tissue do you think will you take? Which tissue will be easiest to get?
Chick tissue.
What tissue?
Epithelium.
What tissue did you say?
Epithelium.
Epithelium where? From where epithelium?
So cheeks.
In
Cheeks. Okay, that is possible. But if you have, I don't know if you have done this in the experiment. If you have done this experiment of making the slide by obtaining epithelial tissue from from the cheek area, or that is the cheek cell, then I am sure after you make that slide, you won't get too many cells there. Very few cells you will be able to see, something that you can count, and you cannot work with that. If I ask you to donate some of your tissue, which tissue will you be most happy and will be easiest for you to donate?
The hair.
Sorry?
The hair tissues.
Blood. Did you say blood?
So I was saying hair.
Okay, your voice is not maybe very clear. I am not able to hear clearly, but
Sir, he was saying hair.
hair, hair, come on. Hair does not contain tissue. Hair is made up of protein. Come on. That is not something accepted. Hair is not made up of cells. Hair is protein purely, nothing else. So the best, easiest tissue to get from any human being is the blood. From the blood, you obtain the WBCs, that is the blood cells, which contain nucleus. So your first step will be to obtain some cells. Okay, for plant, it is not an issue. From plant, you can get anything you want. It is not going to complain. But from animals, you cannot simply take any tissue you want, especially not from human beings. Then your second step will be to obtain the DNA, whole DNA. You cannot simply take only one gene. So you need some technology. So now obtaining tissue is not something difficult. We all know how we can get blood. But what is the technology requirement, the tools requirement is how you will get the DNA from those cells. So that's why you see the first step is DNA isolation. How will you isolate the DNA? We have it in our practicals also, right? So although we are taking one example in our brain, we are keeping one example, but at every point of time, you should think that we have to look at it in general also. So DNA isolation, whether from plants or from other animals or from bacteria or human cells, there are different techniques of DNA isolation. The next thing is from that DNA which you have isolated, you have to separate a very specific gene. For example, in our example, you have to isolate the insulin gene. How is it possible? For that, two things. One is you should know how to identify the insulin gene. Without identifying, you cannot. Secondly, from the whole DNA, this is your whole DNA molecule, suppose, and in this entire DNA molecule, from here to here, this is the insulin gene. Okay, so you need a technology to cut the DNA here and here so that you separate out the specific gene of interest. We normally use the word gene of interest, right? Then sometimes there is a process of amplification also required. We'll discuss that separately. Amplification means a technology to make multiple copies of that particular gene. Okay, so using a technology known as PCR. PCR is not a very new name in today's world. Even layman knows about the PCR. Word PCR at least, the abbreviated word PCR at least, all of us have heard about, although we may not know. So we'll talk about this PCR. Then we said we will put it inside bacteria. Now the big question is, for bacteria, it is a foreign DNA. Every organism, every cell, even our cell, has technology inside by which it will not allow foreign DNA to enter. Like we have system in our home by which we do not allow any foreign organism, individual to enter. Someone whom we do not know, we don't allow them to enter. But still, we may allow someone whom we do not know to enter on certain conditions. One of the most common condition is if that unknown guest comes with someone whom you know, then it is very easy for him to enter your house. Right? Understand this. If suppose one of your family members brings that unknown guest to your house, then you will allow him entry. Right? So similarly here, if you want to insert or introduce this foreign DNA into bacteria, bacteria will not allow. You need someone to carry it to the bacteria. whoever carries is known as a carrier, or another word we use for such a carrier is vector. Now this vector is usually a DNA molecule. Most commonly used vector for bacteria is a plasmid vector. Recall class 11, you have learned in the chapter cell that bacteria has two types of DNA, chromosomal DNA and plasmid DNA. So that plasmid DNA, the discovery of plasmid DNA itself is another, you can say, a great discovery without which probably biotechnology would not have been possible. Right? So plasmid DNA is the most commonly used vector who acts like a carrier. So your foreign gene you add to this vector, and then this vector go inside the bacteria carrying your foreign gene inside. Okay. Then also it is not easy to put inside the bacteria. You need some technology where we call it as making a competent host cell. Like a very simple example, even if your, let's say your family member has brought someone with him or her to your house, but what is the first thing required to enter your house? You have to open the door, right? If you do not open the door, how will anyone enter? Okay, so same thing, the bacteria has to open the door for the foreign molecule to come inside. Although it is brought by a plasmid, plasmid is something which is known to the bacteria. So bacteria will allow the entry to the plasmid who brought the foreign DNA along with it, but bacteria has to open the door. So that is another technology required. How you make the host cell, bacteria which is a host cell here, how will you make the host cell competent to be able to take up the foreign DNA? Right? After that, there are two possibilities. This part is not in your book. We'll discuss about it later. But taking the example of insulin, this is the next step where you obtain the foreign gene product. In our example, what was that gene product? Insulin is our gene product, which is a foreign gene product. Why you call it as foreign gene product? Because it is produced in bacteria. For bacteria, it is a foreign gene product. And then a very, very important last step called as downstream processing. Downstream processing is like purifying the entire product and make it user ready. Okay, something that can be now ready to use. Okay, so that process is called as a downstream processing. So whatever, in short, these are the steps required. So you understood that you need, at the first step, you need something to cut the DNA here. Okay, so you need some kind of a scissor. In fact, we call it as a molecular scissor which can cut the molecules. Another technology is PCR, how you can amplify the gene if necessary. Then how you will do the ligation. Ligation means joining with a vector. Vector is again a tool. Vector is therefore a tool by which you can introduce. Then there are some technology by which you make the host cell competent. Means host cell is made capable to open the door to the foreign gene. Okay. Did you people get some initial, at least some initial? I'm not expecting you to understand 100%, but at least some amount of initial brief understanding that what is normally done in a genetic engineering technique. If anyone has any questions, please tell me now.
Sir,
Yes, tell me.
sir, for competent host cell, sir, is it a matter of finding a competent host cell or modifying a cell to be competent?
When we learn about the technique, you will understand there are techniques by which we make the host cell competent. There are techniques by which, like for example, it's not about finding a host cell, it is about making the host cell competent or capable to take up the particular foreign DNA. For example, in bacteria, we do something called as heat shock treatment. When this heat shock treatment is done, then there are small pores which are formed on the walls of the bacteria, and that is transient pores, means these pores will remain for a very short period of time. So when these pores are formed for just few seconds, within those few seconds, the foreign DNA from its external medium will enter inside the cell directly. Okay, so these are techniques by which host cell will become capable to take up the foreign DNA.
Okay, sir.
Anyone else, anything? So with the basic understanding of the steps, we will go into the details of these steps again in future. But before we start talking about the details of these steps, we have to first understand about the tools. Okay. So first type of tool that we talk about is restriction endonuclease enzyme, which are very commonly known as molecular scissors. Okay. These are called as molecular scissors. Let me discuss separately. Are you able to see the whiteboard here?
Yes,
Yes.
sir.
Yes, sir.
Okay. So we call it as molecular scissors. Okay. Please understand. Now, we start from the name, that is enzymes. These are enzymes which can break down the DNA molecule. These enzymes are known as nuclease enzyme. Okay, basically they are the enzyme which can cut what? Nucleic acids. Because they can cut nucleic acid, so we call it as nuclease enzyme. Now these nuclease enzyme are of two types. One is called as exonuclease, other is called as endonuclease. Okay. Now these endonuclease enzymes are those which can cut the DNA inside, because we know that DNA is basically nothing but it is made up of many nucleic acids. So if I draw something like this, like you will get the DNA something like this. It's a polymer of nucleic acids like this, right? And it is made up of two strands. So these are two strands we are looking at. These blocks are what? Nucleotides, right? And we also know that between nucleotides there are bonds, like these are hydrogen bonds. Okay. So first of all, nucleic acid, what does it do? Nuclease enzyme, what does it do? Nuclease enzyme is responsible for cutting these bonds. What are these bonds? Which join the adjacent nucleotides. Because if you want to cut it, break it down, you have to break this bond, you have to break this bond. What are these bonds? Phosphodiester. Phosphodiester bond. So actually it is responsible for cutting these phosphodiester bonds. Now when we say exonuclease, exonuclease are of two types. We call it as 3' exonuclease and 5' exonuclease. You know that the two strands have polarity. If one is 3' to 5', other is anti-parallel, so it has opposite polarity 5' to 3'. So 3' exonuclease will cut the nucleotides from the end. Exo means outside. So from the end means it will first cut here. And on the other strand, it will cut here. So if I say 5' exonuclease, it will cut here and it will cut here at the 5' end. But when we say endo, endo means inside, it will cut the DNA molecule inside. Inside means where it will cut, maybe for example here and here. Okay, not at the ends. It will cut the DNA inside somewhere. Now the problem is it may cut inside at a random place, which we do not want. It should not cut at a random place. So after a lot of research, a special type of endonuclease was found to be present in bacteria. Okay, this enzyme was found to be present in bacteria, which is known as restriction endonuclease enzyme. Speciality of this enzyme was that it is able to recognize some very specific sequence. Recognize some specific sequence. For example, you see here, this one. These are lots of examples here. ALU1 is the name of the enzyme. Source, where from it is obtained, Arthrobacter luteus is the name of the bacteria from where it is obtained. One of the most common, let us look at that, EcoR1. EcoR1 is one of the most commonly known example of restriction endonuclease enzyme, obtained from E. coli bacteria, that is Escherichia coli. Recognition sequence is this one, GAATTC in one strand and CTTAAG. Okay, and here, where does it cut? If you see carefully, it will, now this is a sequence which is present within the DNA somewhere. Okay, so it's a long DNA on both the sides. It is a long DNA. Within the DNA, this sequence is present. So this enzyme will cut at this particular point and at this particular point. As a result, you see there is a separation like this. So you see this is one side separated and this is another side separated. So that is how the molecule got cut into two pieces. Okay. See, this is one example to explain how the cutting and joining can happen. Okay, these are two different molecules, white color, black color. So when you cut out the part of white color and cut out the part of black color molecule, you can rejoin them like this. Anyway, this will come later on. First of all, understand that there are different enzymes. These are different enzyme, different sources. They have some specific recognition sequences. So they will not cut randomly here and there. They can identify a particular recognition sequence and then they will cut within that recognition sequence at a particular place. So here I have given two tables of certain examples. However, like that, there are a huge number, almost more than 3,000 restriction enzymes have been studied in detail, and almost 600 or more than 600 are commercially available, okay, which are regularly used for cutting the DNA molecule. Right. Now, when you look at the examples, as far as discovery goes, 1978 Nobel Prize was given for Daniel Nathans, Warner Arbor, and Hamilton Oldsmith, who discovered this restriction endonuclease enzyme. The first enzyme to be isolated was Hind2. In 1970, it was first isolated, right? So these things you can read by yourself and understand. It's no big deal. This is something which is not required, already mentioned as extra. These slides were actually prepared long back for a lot of extra discussions. So that's where we discussed about this type of extra information, where we say that these enzymes are classified into 4 different groups. Okay, out of which most commonly we use the type 2 enzymes. Type 1 and type 3 enzymes are not widely used. Type 4 is also not used. Type 2 is the most commonly used enzymes. Okay, so their differences are given here. This is one thing, how we name these enzymes. Okay, some examples are given, like eco K. Often in many of the enzymes you will see the R, we are not using this prefix R. Okay, we directly say eco R1. Eco R1. This R has a different meaning, but actually there should be an R as a prefix also to be used. But most of the literatures, they don't use it. This R stands for restriction enzyme. Okay. You may not use it also. If you do not want to use it, fine, leave it, no need. But E-co, like here you say E-co. E come from Escherichia E, and co comes from first 2 letter of coli. Then after that, whatever is present, that has different meaning. Like here K is meaning strain K. Okay. Like here R is meaning a plasmid. This is plasmid R. Okay. Similarly, Hind, the D stands for the strain, which is known as RD. Okay. Now, after that, these numerical, Roman 1, 2, 3, 4 are given, like Hind 2, Hind 3. Similarly, you have something called as Eco R1, Eco R2, like that. So these 1, 2 are actually nothing but what happens, one bacteria may produce multiple enzymes. And then in terms of how they are identified, okay, first one identified, you call it as 1, second one identified, you call it as second, okay, and so on. So you can see Hind 2, it is Haemophilus influenza strain RD, but 2 means second identified, 3 means third identified. Similarly, Eco R1, it is Escherichia coli plasmid R. First identified 1, second identified 2, and so on. How many ever enzymes are there, they will be represented like that. So this is about the naming, how the nomenclature is done. Because I gave you the list of different enzymes. So in those different enzymes, how the naming is done, this is the first thing. Okay, you can see ALU1. So ALU comes from, A comes from Arthrobacter, LU comes from luteus. Okay, then 1 is the Roman numeral. Then BAM H1 is BAM, B comes from Bacillus, M comes from amyloliquefaciens. Then H is the particular strain, and 1 is, that is not I, that is 1. So 1 is again a sequence number. Like you can see Eco R1, Eco R2, Eco R5. Okay, these are all given here. Now, you see the, yes, tell me.
I can see your, are you sharing the screen?
Yeah, my screen is being shared already. Are the others able to see?
Yes, sir.
Yes, sir.
If you are unable to see, just leave and rejoin. There might be some network issue. Okay, so if you see the recognition sequences, very carefully, if you see recognition sequence, most of the recognition sequences have a very typical design. Can anyone identify recognition sequences? Can anyone identify how the design is very specific? Anyone? Come on. No?
Sir, it's reversed.
Huh?
Sir, is it reversed?
See, recognition sequences, for example, this is echo R1 recognition sequence. If one strand you read, then it comes as GAA. Here it is. Fine. Let me just write 1 second. Did the code go? So if we look at the recognition sequence of echo R1, then what you will see is one strand is 5 dash G A T T C 3 dash. This is one strand. Okay, so obviously the other strand will be 3 dash C T T A G 5 dash. So if you read one strand from one particular side, 5 dash to 3 dash, always you read 5 dash to 3 dash, it is GAATTC. Other one also you read 5 dash to 3 dash, it is same GAATTC. So if you read from both the side, you will see that they are of the same nucleotide sequence. Okay, from both the side they appear of the same nucleotide sequence. This one GAATTC and GAATTC. Okay, now what happens is it comes like this. When a particular strand, if we say GAAAAG like this, when we see, then when we break it from the center, okay, we see a mirror image of each other in a single strand. Okay, the first half is a mirror image of the second half. Such a sequence is called as a palindrome. It's called as a palindromic sequence. Okay. Same thing if I convert it into double strand. So complementary sequence, complementary strand will also become CTT-TTC. That is also palindrome. Okay. Now, in the second half, out of this part, if I reverse this sequence up and down, means TTC goes up, AAZ comes down, then you will get this part, TTC above, AAG below. This is called as a palindrome with rotational symmetry. This is not given in your book. This is called as a palindrome with rotational symmetry. In your book, they have simply spoken about a palindromic sequence. So the recognition sequences of these type 3, type 2 restriction endonuclease enzyme are usually palindromic in nature. Okay, remember that's important. So you see here 5-AGCT on one side, this way also AGCT. Okay, then the second one, if you see for BAMH1, GGATCC and other side GGATCC. Correct? So like that you will see. Next thing to understand is that when they will make the cut, okay, when the cut is made, there are two types of cut which are made. When we look at echo R1 cleavage, this is the recognition sequence. First one is recognition sequence of echo R1. Echo R1 will recognize this particular sequence anywhere in the DNA, and they will cut in such a way that first cut, these arrows did not fall on place. First cut will be here between G and A, and the second will be between A and G. As a result, you can see the cut is made like this, right? This is one end and this is the other end. These ends are called as protruding ends, or we also call it as sticky ends or cohesive ends. Cohesive means sticky. Why do we call it a sticky? Because when you bring them together, then because these are complementary, you can see A and T complementary, A, T complementary, T, A, T, A. These are complementary sequences, complementary bases. So if you bring such two ends together, they will automatically get attached to each other. Okay, they will automatically get attached. Similarly, another example of NLA tree is given. Here it is cutting at the beginning of the sequences here and at the end of the sequence. So here also protruding ends are formed. Clear? Here also protruding ends are formed. But these are some sequences ALU1 cleavage site. What do you see? Their sequence is AGCT, but they cut in such a way that they will form blunt ends. These are called as blunt ends, or sometimes also called as even ends. Right? They don't have protruding ends. They are not sticky in nature because of the cutting position. In both the strands, they are cut at the same position. Here at both the strands, they are cut at different position. Here both the strand, they are cut at the same position here and here. Right? Protruding end has a benefit. What is the benefit? Protruding end benefit is shown in this diagram. This is a molecule A and this is molecule B. Okay, so here eco R1 is cutting in such a way that this part gets removed. So this part is removed. And here molecule B, this part is removed. Right? So as a result, the remaining sticky ends TTAA and here AATT, they are complementary to each other. So as soon as you bring them near to each other, they automatically binds to each other because of hydrogen base pairing. Correct? So this becomes easy. You can easily join two molecules. Correct? That's why protruding ends have a benefit over the blunt ends. Is it understood about the protruding ends, the cutting, how it is done, naming of the enzyme, how it is done, characteristic of the recognition sequence, how it is. Okay, these are the things you need to know about. This is same, another diagram is shown to explain the same thing. Fine. Lastly, a little bit, let us understand that why the particular name of the enzyme is restriction. Endonuclease enzyme. Endonuclease means what you understood. But why the word restriction is used? Many a times commonly just restriction enzyme is also used. Why the word restriction? What is it restricting actually? So this enzyme actually has a different story. What is the story behind? Now we know that this enzyme is naturally found in bacteria. So the question is, what is it doing in bacteria? Right? What is its function in bacteria? We are using it for some purpose. That's a different story. But naturally in bacteria, what is it doing? So understand, inside the bacteria, it will selectively cut up foreign DNA. Okay? Foreign DNA means what could be the source of foreign DNA? Most commonly, this foreign DNA is from the viruses, bacteriophages, which will infect it. Therefore, it will restrict. What does it do? It restricts infection. It will not allow the viruses to infect it. So can you say it is a kind of immune system for the bacteria? It is the immune system of the bacteria. Okay, so bacteria will not easily get affected by the viruses. Right. Next is, this is one important point. What is its function in bacteria? Immediately, it should also come to your mind that bacteria is a prokaryote, which means its genetic material is not covered by a nuclear membrane. It has a naked genetic material. Its DNA is exposed. Where is the enzyme? Restriction enzyme. It is in the cytoplasm. Now, it means that DNA is exposed to this enzyme in the cytoplasm itself. In that case, this enzyme will break down the bacteria's own DNA also. Right? It can break down bacteria's own DNA also, because bacteria does not have a covering of genetic material, and the same cytoplasm, it has DNA as well as the enzyme also, but that does not happen. So how is bacteria's own DNA protected from the function or action of this enzyme? So for that, host DNA, that is the bacterial DNA, has a system called as methylation, addition of a methyl group. Recall the structure of adenine and cytosine. There you have a facility to add a methyl group, 5-methyl cytosine, recall. So that methylation, when it is done, then the enzyme activity fails. The enzyme fails to identify the DNA or to cut the DNA. So its own DNA, it will carry out a modification by carrying out the methylation. However, foreign DNA will not have the methylation. The specific methylation will not be there in the foreign DNA. So it will automatically cut the foreign DNA, its own DNA it will not cut. So this entire process is together known as restriction modification system. So restriction endonuclease enzyme in bacteria is responsible for this restriction modification system. This was the natural story of this enzyme in the bacteria. And when we found that this enzyme has a speciality, what is the speciality? That it can identify a particular sequence. So now, go back to our example. What did I say? This is our DNA molecule. Suppose my aim is I want to cut the DNA, this part I want to separate. So what is necessary? Means this region and this region, the region immediately outside the gene of interest. What is the sequence of this region? I should know. Okay, and I have the database. I have a database like this. Huge database is there, where I have to find which enzyme can be used to break down this, to identify which has that specific recognition sequence identification. Okay, let us say, for example, in this region and in this region, what do I identify? I see that there is a GAATTC sequence. So immediately from database, I come to know that GAATTC is a recognition sequence for EcoR1. So what I will do, this DNA I will be treating with EcoR1. So immediately it will cut here, it will cut here, and my gene of interest will get separated. That's how I can separate my gene of interest from the DNA molecule. That was about the restriction endonuclease enzyme. Is it clear for everyone? Anyone has any question? That was all for today. If anyone has any question, please tell me.